The broadcast is now starting. All attendees are in listen-only mode.
Hi, everybody. It's good to quote unquote see you today. I wanted to welcome you to the fifth in our series on risk-based monitoring on central monitoring practices. I wanted to also give you some pretty exciting news. We went through all of the attendees, and there are 61 of you that have been at multiple sessions, and some of you have been at every one. So I feel like certainly you know me, and I hope I'll get to know you better. And I appreciate any feedback you guys can give. Our webinar series finishes on February 19th. And if you have other webinars you would like us to give, please let us know, because our goal is education, and we're really excited to help you on your journey. So let's get started. Some of you have heard this before, but just to make sure everybody is in the same place. Why is MANA RBM the one that is providing this webinar? And the reason is that we've been focusing on the principles of RBM and remote trial management for more than 10 years on different aspects of it. I'm a physician. I have been trained. I was a PI at the NIH. I've been a vice president at GSK. I've done major change management initiatives, and I've also been a chief medical officer at 2 technology companies. So I have a really broad background and a broad range of expertises, which has allowed us to really expand on what we can do in risk-based monitoring. My team members have worked with me to implement this MANA method for risk-based monitoring with multiple sponsors. And in addition, we've also completed a knowledge transfer to a sponsor. As I said, we've done multiple trials, and in addition, we have also developed with a group of experts, and we've also published competencies for site monitors on risk-based monitoring. So our goal is to expand in the industry and to teach you. As you know, some of you know, our videos are available on our YouTube channel, MANA Risk-Based Monitoring. You can subscribe today and you'll know when the new ones are updated. Last week's lesson 4 or webinar 4, which is on lessons learned, should be updated either today or early next week, as well as this video. Also, if you would like updates on our new content, on blogs, on any of the other materials, please like us on Facebook. It's MANA RBM, Risk-Based Monitoring, and you can also follow us on Google+. So lots of ways to get in touch with us, as well as our website, MANA RBM. We will be launching a new website probably Monday, and we have some additional content on there that should be useful to you as well. So our goal is to help you with anything related to RBM so that we can expand the knowledge in the industry. So we have some exciting news that I thought I'd share with you. First of all, we just found out yesterday that we were named as one of the finalists in the Clinical Informatics Best Practice Award. And that award recognizes outstanding examples of applied strategic innovation, partnerships, deployments, and collaborations that manifestly improve the clinical trial process. We were awarded, or we were named a finalist because of our knowledge transfer with a company called PaxVax in California, and we're very, very excited about that. I will also be releasing, probably on Monday, a blog on what C-level executives need to know about RBM. So stay tuned. As I said, you know how to reach us in all of the ways so that you can be connected and know what's going on and what we're developing. For this program, the goal of the program is to help you to understand risk-based monitoring principles, goals, and how to achieve those goals. And as you know, for each webinar, we have a series of learning objectives. For today's, they include that the monitor will be able to describe how the site monitor, the data manager, and the central monitor interact. It's such a critical piece for implementing risk-based monitoring. We really want to focus on that. We want to describe the type of oversight that the central monitor conducts, and then we want to describe how the site monitor and data manager's input is critical to the central monitoring process, because each team member plays an absolutely critical role, and what we want to show you is how they all go together to get the ultimate goal of better quality for your trials. So a little bit of background and making sure, because we have new people on the webinar this week, is let's go through some process reviews. So remember, the first thing we want to do is risk assessment and categorization. The document many people use is a RACT. It's called Risk Assessment and Categorization Tool. There is one that was released by Transcelerate, but you don't have to use that. You can develop your own. You can make it very simple. Some of our, if you want more information about that, go to our webinar on risk assessment, and there are actually two of those because we ran over. The second is that you're going to define your high-risk data and processes. And the document that will come out of that is usually a specification for collecting data and developing reports. So it really will usually be in your data management plan as to where, what data are going to be collected to support the assessment of these high-risk areas, as well as GCP, et cetera, and the reports that will be there and how those reports will be developed. And finally, you need to determine who will review what and when. So what will the data manager review? What will the site monitor review? What will the medical monitor review, and the central monitor? And as I mentioned to you, we use a data and document review guidelines, mainly because this is a document that has to go across functional groups. So we want a document that doesn't sit, we want this information not to sit in different places because it's too hard to update, and we want it to be a document that everybody can access. So that's why we've developed this approach, and then we refer to the data and document review guidelines in our monitoring plan, data management plan, quality management plan, etc. And finally, completing review, and that's what we're talking about this week and next week. So we're going to talk about this week the central review because it's different and most people don't know about it, and then next week we're going to talk more about the monitor review, the source data review. So some of you who have been on our other webinars recognize this picture that basically this is traditional monitor is done by data sets, and it's done only SDV, or source data verification. Transcelerate, and I think the regulators believe that monitors should be doing more source data review, and as I mentioned, we'll talk about that more next week. And then our central monitor does these two levels of review. So they do the site performance across subjects, which identifies systematic site errors, and then they do site performance across sites. And this really identifies outlier sites or outlier areas that you need to focus on and understand better. So this is what we're going to focus on today. Now, just to go back a little bit to give you a little bit of context, many of you have heard the term of a triangular review, that you look at the medical history and make sure that there is associated concomitant medications related to that, that if there are abnormal labs, abnormal vital signs, abnormal ECGs, that there are related adverse events entered for that, and if necessary, any concomitant medications are entered, and that if there are abnormal labs at the beginning of a study or vital signs, that those are captured in the medical history. So this is the kind of traditional approach. We'll show you some more comprehensive approaches next week, but just to remind you that a lot of this is not really different than what you've done in the past. Now, another thing that's different in risk-based monitoring is the timelines for review. So just as a little reminder, this was a slide that I've shown before, but if you look at the green bars, this is looking at the time from ECRF entry, not necessarily even subject visit, but ECRF entry to data management query opened. And you can see that that time has ranged from about 90 days to maybe 80 days. In some cases down to 60, that's great, but at least in 2012, it had gone back up to close to 80 days. So this is too long. And so when we talk about our process for review, this is going to be very different, and it's shown here. So just to remind you, from a traditional serial process, that's one person does something and then somebody else does, the site sees the subject, enters the data into a paper source, then takes that paper and transcribes it into EDC. And then finally, the monitors go out to the site and do SDV and informed consent review. And finally, the data managers review the data. And so this can take, as you saw from the slide before, 6 to 12 weeks. Now, with MANA's approach, the MANA method for RBM, what we do is the site, and our preference is the site to enter the data directly into the data collector so that there's no transcription step. This aligns with the FDA guidance on eSource. And then within 5 business days, our data manager and monitors are reviewing that data, as well as documents like the informed consent, so that we can find errors very quickly. And then, and that's a very important piece, because it's the information from this that allows the central monitor to do the central monitoring, which is done monthly. And I say monthly because that's kind of a ballpark. If you have a study where you're reviewing subjects much more, or enrolling subjects more slowly, this may be a different time point. But just to give you kind of a context and a relationship between the site monitoring, the subject monitoring and the central monitoring. So let's go through this again. We've shown this before, but now it's going to start. I think now this is probably starting to all make sense to you. So we use our tier 1 as our DM review. So they don't wait anymore. They start right away. And what they're looking for is missing data, missing assessments. The key efficacy endpoints, are those being collected correctly? The dosing, is the dosing being done correctly? Now, and then the monitor is also reviewing that data really at the same time. So the monitoring review is pretty similar to what you did before, but you're doing it remotely. And the reason that we can do this, one is because the subjects are entering, or the sites are entering data directly as much as possible. And the second piece is, we know from the literature that the very little data changes with SDV. So we're eliminating most of SDV, not all of it, but most of it, and focusing on this different review. So in that, we're looking at enrollment, we're looking at inclusion, exclusion criteria, AE, ConMed, med history labs, this rectangular review we talked about. Doing the source data review using our subject profile, analyzing risk. And this we're going to talk a lot more about next week. GCP compliance, document review. So remember, we want to use electronic investigator site files so we can do the informed consent review right away, that we can do any other review of documents that might be needed right away as well, that we can look at the IP management and reconciliation, and we can look at safety, whether it's labs or whatever. So these two are happening pretty much contemporaneously. And the result of that are these in yellow. So if there are any deviations, we would like those entered at the right away. Now, some sponsors take the deviations and they don't enter them in the EDC, or they do a combined deviation report, and you get that at least monthly. That's fine too. Again, but what doesn't work well is if you wait to do deviations, to find deviations till the end of the study. And this is, I've worked with companies that have done this, and this is, I think, a real risk to your quality. If you don't know what the deviations are, then you can't really respond to those. You can't be proactive. So I highlighted this because capturing those deviations, and those are deviations both at the subject level and any other deviations for the study, that information is critical to this Tier 3 review. The other aspects of the Tier 3 review are, data on enrollment rates, screen failure rates, safety, so what the AEs are, what the SAEs are, the areas of premature discontinuation, and you may remember from previous webinars that we talked about that this is an area that the FDA looks at when they're deciding who they're going to do for their audit. So again, they're looking at is this an unusual rate? And then queries, so items with a high frequency of queries, sites with a high frequency of queries. Now, in addition to all of this, which really comes out of the Tier 1 and Tier 2 review, the central monitor is also looking at variability of data across subjects and the things like timing of assessments. They're looking at things in our issue management system, as well as looking at monitoring reports. So you can see that all of these things really fit together, and that's an important piece. So if the monitors don't do their monitoring, if the data managers don't do that right away, then that data is not really available for the central monitor to do their oversight. So I really want to emphasize this kind of synthesized, integrated approach. And that's what we keep emphasizing, is that this is a cross-functional effort. So now let's look at some of the cross-subject evaluation. And this is an assessment that, these are assessments that may not have been done ever before, but what we're looking for are really, here we're looking for evidence of fraud. That's the basic thing. So are all the vital signs for a specific visit, that's across subjects, being done at the same time according to the measurement times entered? Are all the vital sign measure, and I'm using vital signs, it could be an ECG measurement, it could be a peak flow, it could be any of the assessments that you have that you're seeing across visits. So are these measurements for a specific subject done at the same time at every visit? So what we're looking for are ways that might be evidence of fraud, or that the site is systematically making errors. It could be either one, but that's what you're looking for. So these are assessment times, assessment measures. Values, are the vital sign measures done at time points that end in 0 or some other number, 5 or 3 or 7? Are the vital sign measures the same across time or have the same ending numbers? And I'm going to show you an example here. And then an evaluation of study staff performance. And remember when I did the very first webinar, we talked about J&J and Icon having a 483, of where a specific person was giving infusions or injections, I can't remember which, but they gave that same activity at the same time for multiple subjects. So what you're wanting to do is line this up so that you can see, is that something that's happening? Because again, it's a systematic error or it's fraud, and that's what we're looking for. So one of the things that we think is valuable is to think about the data that you might want to be able to do these assessments and make sure you're collecting it. So for instance, traditionally in your EDC, you don't collect the initials of the person doing the activities. That's not an analysis field. But if you're wanting to look if that person did that activity at the same time for multiple subjects, you need to know that. So that's what I talk about, thinking about what it is you want to look at when you're doing your data and document review guidelines, and making sure you've collected that information in your EDC or wherever. But you can't run reports if you don't have that as a data field. So this is a really, really important point. So let's look at an example. And I use this example because this was the one, I showed this to you before, but this came from Jonathan Helfgott at the FDA, who at that time was, I believe, head of the biological, basically the monitoring group or the audit group of the FDA. And he presented this at a program that I was at. So again, this is blood pressure values, and it was an important secondary endpoint, and it was important for evaluating risk benefit. So this is, if we go back to our high risk data elements, if it's important for efficacy, if it's important for safety, it's important to look at. And what they saw was the lack of expected variability in blood pressure measurements. And if we look at this, you can see what I mean. So this is multiple measurements. So this is the same subject, and these are all the different visits, okay? And these are the dates of the visits. Now, had I done it, I would like to also collect the time of the visit and who did the assessments, just to be additional checks that you want to do. But again, this is a simple report. So sometimes you'll see a lot of emphasis on statistical methods, and yes, you can use things like analysis of variance to do this. But the other thing is you can just make a table and look at it, and these things pop out. So if you look at this, how often, those of you who are from the sites can answer this very well, but everybody can, how often do you get exactly the same blood pressure when it's measured twice? Very seldom. And then if you look at this, you can see that there's an awful lot of zeros, a few eights. You can also see that the same measure on both assessments, unusual, almost impossible. And then if you look at the measures on these two dates, these are two visits, very different, and they have, excuse me, little jumpy there. They have exactly the same measurements. This says there's something fishy going on. So this is why you want to find these out before you have your site audited. This is exactly what we're talking about looking at with a cross-subject review within a site. And you wouldn't pick it up well, you might have picked this up if you're doing vital signs on a specific CRF, but you clearly wouldn't pick it up from visit to visit, because that's not something as a monitor you would even notice, because you're looking at the vital signs from one visit and then you go to the next visit. So it would be very difficult to pick up if you didn't plan this kind of a review. Now, I'm going to let you look at this data and we're going to do a poll. So let me just walk you through it. These are multiple subjects at one site, and this is an AE review. So these are the terms. This is the day of the first dose, start date, severity, whether it was an SAE, and when it was reported. Now, let's go to our poll, okay? And the question, select one. What trend do you see in the data provided? Okay, and you have three choices. There are 3 SAEs, but they are for different things. There are 3 SAEs, but they may be similar, or there appears to be a common AE finding of skin reactions. So if you can go ahead and answer the poll, oh man, you guys are awesome. So far, so good. We only have 22%, so if you'll keep voting, I'll give you a few more seconds so you can think about it. It's hard. I'm sorry the poll is covering. I can't move it. That's the problem. I can't change it. So we'll just have to, hopefully you got enough chance to look at it. So we give you 5 more seconds. All right, we're going to close it now and we'll look at the results. So 4% said there are 3 SAEs, but they're for different things. 12% said there are 3 SAEs, and that may be similar. And then 84%, there appears to be a common AE finding of skin reactions, okay? So let's go back and look at this as a team, okay? So here we go. You're right. So there are a lot of subjects. They do have different things, but they do appear to be common. So, and this was an area when I was a medical monitor, I was an expert on rash. But so you can see that they have different names, but they're clearly all some kind of skin reaction. And even though some of them are SAEs and some of them are not, that doesn't matter, because it doesn't matter the severity. What you're really wanting to see is, do these things look common? And what you're seeing here is, this is clearly something going on with the safety of this study. And you might not have, again, picked it up if you didn't look at that data altogether for all of the subjects at a site, or we would also look at this at a central level as well. So that's why we want to make sure that you think about how you look at the data to give you a better idea of what's going on. Now let's switch to cross-site review, because that's where you're going to spend a lot of time. So this is what I call your most important pearl. So remember this, screenshot this, whatever. This is really important. So one of the things that I emphasize over and over and over again is the importance of normalizing data. And what that means is it allows you to compare data from one site to another. So to do that, you need to normalize the data based on some criteria. So what I did is to take some of the things that we look at and explain that you don't always use the same thing to divide it by, for instance. So for instance, when you're looking at number of informed consent deviations, you know every subject gets an informed consent. So you want to use every subject or your total of all subjects enrolled. And then that way you can get a deviation rate, in fact. So you're looking at the number of informed consent deviations divided by the number of subjects enrolled. You wouldn't use the subjects randomized because the fact of the informed consents are really collected for every subject. Now, in contrast, your dosing deviations are different because you don't get dosed unless you're randomized. And again, I'm using these terms generally. They may be a little bit different, depending on your study. But let's just, in general, if you're randomized, you receive treatment with the study investigational product. So if you have dosing deviations, you wouldn't see those in all subjects enrolled. You wouldn't see those in the screen failures. You'd only see those in the subjects that were randomized. So that's why you would use this to normalize your dosing deviations. Now, we're really evolving on the normalization of AEs. So if you're doing a short study, so let's just say it's a phase 1 study, or it's a study that you're only going to follow patients for a month, something like that, then it's fine just to use the subjects randomized. So the number of AEs divided by the subjects randomized, because they're going to be the ones getting treatment. But if you're doing an oncology study, let's say, where you may have patients treated for months or years, then you probably want to normalize that based on the duration of treatment, a number of study days, number of study days on treatment at the site, or something like that. So you need to realize that the normalization you do around something like AEs may be different depending on whether it's a short study or a long study. And that may go for other areas too. I'm just using this as an example. Now, the other thing that we have found tremendously valuable, and I think our team loves, is when we're looking at things like onset of AEs, or we're trying to compare different assessments, we like to normalize that data either by study day or what's called relative day. And what that means is you're looking, so day 1 is usually the first day of dosing. And so you can then compare everybody that was dosed, based on how many days of treatment they had. And this allows us to look at data in a different way and to find trends sometimes that we wouldn't find otherwise. So this is your pearl. Don't forget this if you're thinking about how to design your reports for cross-site review. Now, I've got a few polls in here for you today. So here's another. First, we want you to look at this. So what we're looking here is at the number of AEs per site, okay? And we're going to go in and we're going to do another poll. Okay. And I'm afraid it may go over, so take a look at your data, because I'm afraid the poll may roll over this. So here's the thing. So what sites have a problematic rate of AEs? Is it site 6, site 5 and 6, or you don't know because you have to normalize the data? Okay. Ooh, you guys are going fast. Okay, I'm going to give it just a couple more seconds. We'll go to 30 seconds, and then if you vote, okay, 2 more seconds, we're going to close it. And let's see the results. Okay, nobody thought just site 6. 24% of you thought site 5 and 6, and I totally agree. If this was the only table you had, that would make sense. But what we just learned is that you need to normalize that data by the number of subjects treated, and 76% of you got that correct. So let me take this now and show you the results for the randomized, because it's really interesting. I love data. So here you can see, here, let's look at site 6 and site 5, because those were the ones that, clearly had the highest rate of AEs. Now what we want to do is we want to know that once you've normalized that data based on the number of patients that were randomized, which site actually popped up? It was site 8. And if you look here, they didn't have very many AEs, but they had fewer patients that were randomized. So the number of AEs per subject actually went up. The other thing I want you to notice, and again, is that in this, I want you to notice the axes, because those can kind of fool you, and depending on the reporting tool you have, they may look differently, and they should, because you're wanting to have the axis fit with the data. So the bottom here, the axis is the site number, and here is the number of AEs per site. And you can see here that site 6 had 85-ish, but when you corrected it for the number of randomized subjects, you see they had 6 and change, maybe 6.5 or whatever. But site 8 had 12.5. So if you hadn't randomized that, hadn't normalized that data, you would have missed the most important data trends. So that's why this is really important. Now I'm going to give you this is kind of some lists, but I want to make sure that you think about these, that you can remember these, because these are going to be common to pretty much every study you do. You're going to want to know that. So, and a lot of these are the data. We went through this, I can't remember which of the webinars, but what the FDA looks at when the information that they want to decide where they're going to do their audits. And they want the enrollment rate and the screen failure rate for each site. And the screen failure rate really helps you understand whether the site is understanding the study correctly, or maybe whether they're wanting to screen a whole bunch of patients so that they can get paid for, but whatever. These are patients that probably shouldn't have been enrolled in the study. Now, you do know that screen failure rates vary across studies, and so sometimes you're going to expect a high one, but you want to look at for anybody that's an outlier. And remember, your screen failure rate must be looked at in the context of enrollment. Now, premature discontinuations, another red flag, because the FDA looks at it as these may be unreported AEs, so they want to know how many were lost to follow-up, how many had an intercurrent illness. All of these they see as potential safety signals that were missed. And so you want to very carefully look at your premature discontinuations. Protocol deviations. Remember protocol compliance, when we talked about that, the FDA and the IRBs are doing their risk benefit based on the protocol. So if you're not following or the site's not following the protocol, that's increasing risk. So you're wanting to look for any deviations around inclusion, exclusion, dosing, missing assessments, really all of these. Now, I bring this up because there are both subject deviations, but there are also non-subject deviations. So it may be the site didn't store their drug right, or the site didn't, the pharmacy didn't reconstitute the medication right, or there could be any number of different things that are deviations from the protocol, and they're not all subject related. So one of the things that we do, and we'll talk more about that on the February 19th program, is we also capture non-subject deviations somewhere else besides the main study, so that we can combine these, because we're wanting to look at the total protocol compliance for the study. Also, we want to look at any AEs that were not recorded based on lab abnormalities, ECG abnormalities, vitals, or any other safety findings. We want to look at randomization errors. So this is something that this may be done by an unblinded statistician, but you want to look at were the subjects randomized in order. So you want to look at things like the timing of the randomization. Were they given the wrong medication per randomization scheme? And we all know that happens. Or were they given the wrong dose? So they were randomized correctly, they gave them the right treatment, but they did the dosing wrong. So all of these would be things that you want to look at. Also, any other dosing errors. Were they using the wrong dosing regimen? Were they storing the IP wrong? Under documentation, I listed informed consent errors, but there are other things as well. So if you have local labs, if you have other source that you might want to have, any of that documentation, you want, notification of the IRB, all of that, you want to make sure that you're capturing and looking at it as the context of how that study is running. And finally, don't forget your high-risk data elements and reports. So if I think about it, all of the things up to here, you're going to do for every study. But then for here, you're going to do it only for the high-risk data elements. So this is going to be different for each study. All of these, you should pretty much do the same for every study. Okay. Now, areas to assess. The other thing to think about is if the data are too good. So if you don't, if you see those that are on the high end or those that are always on the low end, if they don't make any errors, is the data real? Because we all make mistakes. Is it relatively homogeneous? Are there no AEs reported when some should be expected? These are all things you want to think about. You want to have an index of suspicion. And when you're looking at the data. So those are the things to think about here. Now, I'm going to go through this with you because there is a poll on this one too, so that I can keep you guys awake. So now, in contrast to the last one, which was all subjects at a site, these are all different sites. And you can see that these data were normalized. So we've already done that, so you don't have to worry about that. That's not the trick question here. Now, let's look at this. So if you look at site 1, and this is percent of all AEs. So what was the percent, how much did these sites contribute to all of the AEs? Okay. And then what did they contribute to the percent of neuro AEs? Just take a minute and look at it. And what was the percent of CV AEs? So you want to just look at that. Remember, you're looking at the highs and the lows, okay? So let's go ahead and launch the poll. I'm sorry that I can't, I'm going to have to figure out how to do this so you can look at this while you're taking the poll. But I'll read you the poll so that you can think about it while you can still see the data. Which sites have a worrisome rate of AEs and in what area? So here's the choices so you can look at them. Site 1002 for all AEs, Site 1008 for neuro AEs, Site 1008 for neuro and CV AEs, and Site 1010 for both neuro and CV AEs. So I'm going to launch the poll now. You've had a chance to look at the data. And hang on, let me get this. And now now the poll's going. I feel like I should be playing some music while you guys are doing this, but I'll let you focus and pay attention. So 5 more seconds. Okay, we're going to close now. And now let's share the results. Okay, so 7% said 1002 for all AEs. 24% of you said 1008 for neuro AEs, 1008 for neuro and CV, 66%, and 1010 for neuro and CV AEs, 3%. Okay, so let's look at the data now, and I'll share with you how I would look at it. So I don't, there's never a right or wrong, but I'll tell you how I look at it. So what I looked at when I looked at this is that these were pretty well distributed, and there may be something here because it's such a small percentage, but I would say that's a reasonable range. But when you look at the neuro AEs, you guys all picked up that this one is pretty high. So 28%, that's more than a quarter of all AEs came from this, neuro AEs came from this site. And if you look at this, they also had the highest percent of CVAEs. So to me, these two do go together. I could see why you could argue that 15 isn't that different from 13. And so if you argue that, I would say, okay, that's a reasonable thing. You thought about it and you looked at it in context and you made the decision. That's again, there's no right or wrong. This one's pretty obvious. Now, the last question was the site 1010, and the reason I brought that up was the way we talked about it was the low numbers. Now, if these had been the lowest numbers and there weren't any other sites that were low, I would definitely say this would have been something that you should look at. But if you look here, there's a 4, 4, 5. So I would say, this is probably not problematic and this probably isn't either. But you could put it on and say, let's look at it next month and see what the data look like. But that's the kind of thing we want you to be thinking about. Now, let's look, you know that I love z-scores, or some of you who've been in all of my programs know I love z-scores because that's a way of looking at the distribution of data across sites. So let's look at the sites prematurely discontinued. This is what you would normally see, okay? And one of the things I do really like is you see that you're showing it for over months. So you see month 3, month 6, and month 9. And it's nice to see the data over time. So I really like that. And one of the things that, you could say, ooh, this is kind of worrisome, so that you can see site 5 and site 6. All of the sites are seeing more premature discontinuation by by month 9. That's probably to be expected. But let's go ahead then and look at the data if we did it as a z-score. Now, just to remind you, the 0 means that that's the mean, and that if you're a -1, you're 1 standard deviation below the mean. And if you're a +1, you're 1 standard deviation above the mean. So you can see these are probably, everybody's pretty well clustered, except for this site. So you might have picked this up here, but you might not have. You might have said, oh, but you can see here, this is clearly almost 3 standard deviations above the mean. Now the data are going in the right direction, but this isn't a site that you really need to focus on. Now, what about this down here where you're -2 standard deviations? This is a site that is keeping their subjects. So if you're -2 standard deviations, that means you are 2 standard deviations below the mean, and you're doing a good job. You have the lowest rate of premature discontinuation. So one thing you might want to be doing here is saying, how are they able to keep their subjects in the study, and to learn from that and share that. So it's not always who are the problem sites, but what can you learn from the sites that are being successful? And we do that all the time with something like enrollment, but let's think about other really, really important things like premature discontinuations.
Penny, we can only see the poll right now.
What? You can only see the poll?
Can you show us?
Oh, I'm sorry.
Yes.
Let's go back. Oh, and that was such a good slide. Let me go to the poll. Thanks, Christine. Okay. So we have those results. And then here's the z-scores that we talked about. So you can see these are over months. Thanks, Christine, for letting me know so everybody can look at this. We'll go over it briefly. So we go month 3, 6, 9, okay? And then when we do the z-score, again, you can see that site 7 is almost 3 standard deviations above the mean, and site 4 is 2 standard deviations. And I always say if you're outside of 2, that's where you want to really look. But in this situation, this is a good thing. So you want to be looking at this carefully. And in fact, here, this could be a good thing, or they've already lost all their patients. We don't really know, but it's something that you want to look at. Now, queries again, and I want to give Christine a chance. So I'm going to go just a little bit longer. If we run out, we'll pick some of this up. But remember, you want to look at the questions or areas that are being queried most often. And are there aspects of the study, either assessments, visit windows, that are getting more queries? And is there one site that has more queries or one individual? Now, again, this is why I like eSource, is because the person that did the assessment is entering the data, And that way we can see if it's an individual rather than an entire site that is having a problem. So when you're using transcription, you often can't differentiate whether it's everybody at a site or an individual. And that allows you to be much more focused in your analysis and support. So it's good to know that. So this is just an example. And you can see here that the diary date had, there were a total of 438 queries. The diary date had 154, and the informed consent date had 61. So these are the areas that you would say, oh, I need to look at this more. Are these queries distributed across all the sites, or are they only in a couple of sites? Are these informed consents distributed so that maybe your study has both local IRBs and central IRBs? We had a study once that had, it was a central IRB. The central IRB informed consent had 2 dates on it, so it was confusing. The sites didn't know which date to use, and so there were a lot of queries. But by identifying this quickly, we were able to go ahead and get that corrected within a couple of days as opposed to weeks and months. So this kind of a review really is very useful. And then time to data entry is another one. And again, remember, if you're using eSource, the time to data entry should be the same. So, and if you see something like this, where at visit 1, that on the visit date and time, it says March 1st, 2015 at 0817, but when you look at the audit trail, it shows it wasn't entered to the next day. If you're using EDC, this isn't a problem. This is actually good. But if you're using eSource, this is a problem. So you want to think about the specific tools that you're using. And then if this, in another situation, if you see just a little bit of change, from 1012 to 1013, that's nothing to worry about. You don't need to spend time worrying about that. But remember, the longer time between subject visit and data entry means more risk to the subject because the review is delayed. And I was on a program with a company that used this as their one of their main risk indicators was time to data entry, and they found it very useful. This was an EDC study. So again, don't forget things like this because they can be useful to you as well. Now, so a couple other things that our central monitors do is to review the open issues and their progress to close, to add new issues, to identify, whether a CAPA needs to be instituted. If it's a major issue, it does, and to look at the post-remediation. So we'll talk more about how we do issue management in the February 19th webinar, but I wanted you to see these and know that this is part of the our central monitor review process, as is review of the monitoring reports to make sure that those monitoring reports are meeting the monitoring plan. We also have the PI sign right off on the monitoring report rather than do a separate letter. Anything we can kind of remove as extra stuff to follow up on that we don't think are needed, we like to have clear communication and have that acknowledged by the PI. And then we also want to confirm any issues that were in the monitoring report have been entered into our issue management system on an issue log. Staff turnover and training is another one. We follow this because when you have staff turnover, there's a high risk of errors as well. So, and there's a lot of things that you need to look at around training. And finally, our central monitors issue a study-wide report that covers everything for all the sites, and this allows the sponsor to know the key issues across the trial in all areas. Important areas like CAPAs are listed, and site performance is provided as well as team performance. And then we break that out and do a monthly site progress report for each site. And not only is that, and that's developed by the central monitor, but delivered by the site monitor. And we have had fantastic feedback from the sites when we provide them this kind of really rich content of what's going on at their site. And that allows, this is great documentation for CRO oversight, which is a key part of GCP as well. Now, I wanted to give you a chance to have Christine McKee of our team share her experience as being a central monitor. So Christine has been working with us, about a year, and I'm going to let her talk a little bit about her background and then share with you her experiences here. So take it away, Christine. I think. Did I mute her?
Hi,
There we go.
I was muted. Hi everyone, I'm Christine. I've been working in biotech for about 10 years, a little over 10 years, with the last 8 in clinical, working as a CTA and a CRA, becoming certified and all that business. For the last year, I've had the opportunity to work as a central monitor, and it's been a really cool experience. And to be honest, I feel like I've grown exponentially over this time. And I feel like that really comes down to how much of the clinical process central monitors are exposed to. Um, as monitors, we strive to ensure the safety of the subjects and the integrity of the clinical data. And working as a central monitor, really with risk-based monitoring tools, has really allowed me to work toward that goal more completely and more efficiently than ever. So training for to be a central monitor was an interesting experience. When the role was presented to me, it was a little intimidating. I was not a tech person. Learning new systems was not on the top of my to-do list. I was more focused on getting to the site, reviewing data. I was comfortable sitting down, digging into charts with source document verification and a source document review. And at MANA, Penny really runs a pretty intensive training. She had me build my foundation from the ground up. I was testing eSource, ePro systems, electronic site files, and TMFs, and testing the reports that pulled the data from pulled the clinical data and presented it in a way for us to analyze it. And so that really gave me a good foundation of what I was working with, and at a level that I never dreamed of even looking into. And in unison with training on all the systems, I started learning the processes, how to utilize the reports to identify trends, how to step away from the granularity of the site monitor and the data manager and what they're reviewing, and look at everything really from a bird's eye view. And the key responsibility, how I view it, of the central monitor is really synthesizing those findings into actionable items, prioritizing them, and following them to close. So what I've learned as a central monitor. Penny, if you want to scoot to the next slide. So as a site monitor, you learn really quickly that relationships are really important. And when you go to the site, you want to make sure the study coordinator knows that you're not there to point out and identify mistakes, but you're a source of support. And that's kind of, the central monitor is everyone's teammate. You know, everybody from the project manager to the site monitor to the CTA, and then everyone on the site, they're all pulled in different directions over the course of a study. And everyone's very busy. So I just think that the central monitor provides support in a way that everyone working on the study may not have even known that they needed. And within that support, you have the ability and the responsibility to hold everyone accountable. And I really feel like we're dotting I's and crossing T's throughout the study in real time in a consistent way across sites and over the duration of the study. So one of the biggest differences from site monitoring to central monitoring is really that you have a bird's eye view of the data. While still having access, you can drill down and identify the specific issues and the trends that you've found. And so Penny's really put together a team approach to this. And I don't think I would have thought of this, but working with data managers is really cool on our Tier 3 review, on our central monitoring review. They come at the data from a different angle, and when we bring all of our findings together, we usually have the same findings, but typically a different way of thinking about how the findings affect the study and then the action items that result from those findings. They're typically different, and it really adds great value to our output. So comparing old and new, would I go back? Short answer is no. I really feel like the central monitor rounds out the oversight of the trial, and it's just like a really great part of the clinical team. And so what I like best about this approach is that it just feels like the natural progression of monitoring. It's 2016. We have all of these tools. We have these people with this experience, and it's our job to apply them in our processes. So what do I want my colleagues to know? I saw an internet meme recently, and the quote was, "The most dangerous phrase in the language is, 'We've always done it this way.'" And the best thing you can do for your clinical team is to bring new ideas, challenge the way you typically do things, and make your process better. And I think that that is really a good summary of what the central monitor does, is how can we make this better? Who's doing a good job? Who's not so doing a good job? And how can we all get on the same page and just do a better job? So the central monitor is really troubleshooting throughout the clinical trial, not at the end for study close or for a milestone or to prep for an audit, but in real time. And I feel like the central monitor is a bit like the clinical team's resident auditor in that way. And it's been a really cool experience.
Thank you, Christine. And again, Christine didn't have any special training before she started. She's been a fantastic central monitor. And I just wanted, part of this is to show you that anybody can do this. Not anybody. I mean, we think Christine is a super member of our team, but this is not something that you have to have very special credentials. It's an interest, an enthusiasm, a goal to have the study done well. So, and a real team approach. So I think that's why our team has been so successful in this. So just in summary, the central monitor reviews data across many different aspects of the study. So oftentimes I've seen in risk-based monitoring, they talk about the central monitor determines when the site monitor should go out to the site. We've taken it a little bit differently. We think that the central monitor is really looking at where there are things that can be improved in the performance of either the site or the staff, and that we can focus on that not necessarily by going to the site, but by spending that time that's needed to help and understand why there are outliers, why things are going the way they are. It does not include individual subject review. That's a big difference and a big change, as Christine said. And that management of issues is a key component. So this is something that's not often done, where issues are entered by lots of different people. And as I said, on February 19th, we'll talk more about that. It is an excellent documentation of CRO oversight. Those reviews that Christine provides to the sponsor are wonderful, and it includes both overall site performance and individual site progress reports, which really help in enhancing the relationships with the sites. We're at the end of our time, but I have a number of questions. So what I would like to do is I will download these questions and provide answers to them, and then we will post these on the website, and you'll be able to go in and download those. So I really think I appreciate it when you ask questions. There'll be a survey that goes out that has the link for next week's program. And don't hesitate to email me. I really want this to be something that helps the community. And if you like our webinars, please tell other people about it. And don't forget to like us on Facebook, and then you'll get all of our updates as well. So thanks very much for your participation, and we'll see you next week. Take care. Bye-bye.